Hello, AP Bio. Today we're going to talk about the effect that mutations can have on the process of gene expression and the building of a protein. We're going to talk about a few different types of mutations and look at a couple of examples of how this happens. So a mutation, as we talked about before, is a permanent change to the genetic information of a cell. So we're not talking about a mistake that's immediately fixed in that process of DNA replication. These are the mistakes that did not get repaired and now are part of that genetic code, part of those instructions in the DNA. We're going to start by talking about substitution mutations. So these are sometimes called point mutations because they happen just at one specific spot within the genetic code. There's a single nucleotide change. So one letter accidentally gets switched to a different one. So this is a mistake in the base pairing. Instead of an A going with a T, perhaps an A gets paired with another A in that process of DNA replication. That's a substitution mutation or a point mutation. Depending on how that changes the codon and the amino acid that's put into the final polypeptide, these types of mutations can have a wide range of effects. Sometimes they actually end up coding for the same amino acid. Like we looked at earlier in the week, there is redundancy in the genetic code. And so if the letter got changed, but it still codes for the same amino acid, there's going to be absolutely no effect. The final protein that's built is going to be absolutely identical to what it would have been before. And you would actually never know that you had that mutation, which is why this type is called a silent mutation. If a letter changes and changes the amino acid, that's referred to as a missense mutation. Think of that one as it still makes sense. It still means something, and a protein will still be built, but it'll have a different amino acid in one position of that polypeptide chain. That sometimes can have a pretty big impact because, as you know, the chemistry of those amino acids is what causes the protein to fold the way that it does. And so if it folds incorrectly and it's not the correct shape, it may not be able to carry out its function the way that it's supposed to. The third possibility is what's called a nonsense mutation. If the mutation changes a letter in the middle of a gene to a stop codon, then that protein won't be built completely. It will stop in the middle, and therefore that protein won't even be made. So that's called a nonsense mutation. We're going to look at one example of that. Sickle cell anemia is a disease you may have heard of that's caused by a substitution mutation. And I'm going to show you just a 1-minute video here that explains what sickle cell anemia is and the genetics behind it. And so I want you to listen for the mutation and see if you can figure out if this is a silent, a missense, or a nonsense mutation.
Sickle cell anemia is a genetic disease that affects hemoglobin, the oxygen transport molecule in the blood. The disease gets its name from the shape of the red blood cells under certain conditions. Some red blood cells become Sickle cell anemia is a genetic disease that affects hemoglobin, the oxygen transport molecule in the blood. The disease gets its name from the shape of the red blood cells under certain conditions. Some red blood cells become sickle-shaped, and these elongated cells get stuck in small blood vessels so that parts of the body don't get the oxygen they need. Sickle cell anemia is caused by a single code letter change in the DNA. This in turn alters one of the amino acids in the hemoglobin protein. Valine sits in the position where glutamic acid should be. The valine makes the hemoglobin molecules stick together, forming long fibers that distort the shape of the red blood cells, and this brings on an attack.
So thinking about So sickle cell anemia was an example of a missense mutation. It puts a different amino acid in place. It substitutes a valine for a glutamic acid. And as you can see, in this case, that has a pretty significant effect. It changes the shape of those proteins and how they interact with each other to form those long strands, which deforms the cells and causes them to get stuck in blood vessels, which leads to all the symptoms of that disease. So that's an example of a missense mutation. Another group of mutations are what are called frame shift mutations. These are mutations that we could also call insertions or deletions, where we're adding an extra base or deleting some bases. Frame shift mutations tend to have a larger effect on the final protein because they alter the entire reading frame. Remember, the genetic code is supposed to be read in chunks of 3 letters. If we add in 1 or 2 letters, especially, or 4 or 5, we knock off that reading frame, not just for that amino acid, for that codon, but for every codon that comes after it in the entire gene. So an example of this is the disease cystic fibrosis. Cystic fibrosis is a mutation that involves a transport protein on cell membranes that's made to transport chloride ions. And this can be caused by a few different mutations, but one of the common ones is a deletion of 3 nucleotides. And that's what's shown here. This is the normal sequence. This is the mutated sequence, and you can see this entire codon has been removed. In this case, since it is a base of 3, it actually doesn't change the rest of the sequence. You can see we still have glycogen and valine, like the last couple amino acids are all the same. But we lost that one. We lost a phenylalanine amino acid in that polypeptide chain. And that mutation is actually enough in this case that this protein is not made correctly and does not function. It does not allow chloride ions to go through, which is what causes all of the symptoms of cystic fibrosis. So this is an example of a frame shift mutation. You can imagine how if we only took out 1 letter, then that would change everything after this code as well. And you're going to be looking at some more examples of that as we continue through here. You have 2 assignments to be working on. You have a mastering biology assignment that's due tonight, but you're also going to continue doing some practice with these different types of mutations and actually looking at what happens when you transcribe and translate and the effect that mutations can have on the protein. So there's a Google form where you'll be going through and looking at 5 different examples. You will need your codon chart when you work on this. I did link it in the Google form if you lost yours. But this assignment is going to be due on Monday night. I hope you enjoy your long weekend. Reach out and ask if you have any questions, and I will see many of you in person on Tuesday and the rest of you live once again. Have a great weekend.